Hello, this is Chapter 7, which is Development and Sex Determination. In this chapter, we look at different sex chromosomes, what they do, and what happens if they don't do what they're supposed to do. Your sex, or your gender, is determined by your sex chromosomes. In this diagram, we see that there are X chromosomes from the female and X and Y chromosomes from the male. So if we put this into a Punnett square, a female produces gametes each containing one X chromosome, and a male produces gametes containing one X chromosome and a different gamete with one Y chromosome. If we combine these chromosomes in a normal Punnett square, we see that the X's come together to make this individual, and the other two X's come together to make this individual, and these are female. And these on the bottom would be male because they are X from the mother's chromosomes and Y from the father's chromosomes. And so if you notice here, we have 50% female and 50% male, which is why you have roughly 50% males and females in the world. So we're going to spend a few seconds looking at the Y chromosome. The Y chromosome is a very small chromosome, and there are a number of genes on there, but arguably the most important gene is this SRY gene. And SRY makes a protein called TDF, which stands for testes determining factor. And not surprisingly, if you have TDF, you make testes, and testes can make testosterone. And from all that, you can end up being a male. If, on the other hand, the SRY gene is mutated, then you don't make TDF, and so you end up being female, because you lack the important bit, the sex-determining region of the Y chromosome. So with SRY, you have male. Without SRY, you have female. The X chromosome, remember, is present in females as two X's, and to be a female, you actually need the absence of a Y chromosome and the presence of two X chromosomes. So you must not have a Y chromosome, but you must have two X chromosomes to be a genetically normal female. This diagram over here shows just some of the many genes on the X chromosome, and it's many times bigger than the Y chromosome. And maybe obviously the embryonic gonad here develops as an ovary in females instead of testes. So a female has two X chromosomes, as shown down here. This is a diagram of some female cells, and a male has one X chromosome, as shown over here. These are some male cells. But apparently males can function just fine with just one X chromosome. So even though females have two X chromosomes, you don't need two. So what's going to happen is that one of these X chromosomes is going to be inactivated. It's going to be reduced and condensed to an inactive state. So this inactivation of X chromosome is random. It occurs only in females. And what's happening is it equalizes the activity of X-linked genes in males and females. So here's a male, and we're having all kinds of gene expression from this X chromosome. In a female, we also have gene expression, but we have the potential for a second amount of gene expression, and that would cause twice as much protein as you might need. So what happens is that this chromosome here gets condensed down and inactivated. You don't make this protein. And so you have dosage compensation, whereby males have one X chromosome expressing and females have one X chromosome expressing. This diagram over here shows bar bodies. A bar body is the name given to this condensed X chromosome. So this condensed X chromosome is a bar body. And you can see them on this diagram. There's one here, and there's one here, and there's one here. They appear as dark dots on the inside of the nucleus of a cell. These bigger circles here are the nuclei of a cell, and the actual cell is much bigger. It's out here somewhere. So these are little dark dots on the inside edge of the nuclear membrane of a female cell. So How many chromosomes do you need? Well, you just need one X and one Y to be a male. If you have two X's, you're female, but you only need one, and you can bar body that one extra. So here's another diagram of a bar body. You can see it down here. But what would happen if you have too many X chromosomes? So here's an individual. They are female, but they have 3 extra chromosomes. Now, you only need one X chromosome to be female, so here we can make 4 bar bodies. And here's 1, 2, 3, and 4. And again on this cell, here's 1, 2, 3, 4 bar bodies in there. So no matter how many X chromosomes you have, you bar body the number that you need to get down to one expressing X chromosome. There's an interesting case of genotype versus phenotype, and it's called androgen insensitivity. If an embryo is XY genotypically, it should be producing testosterone and develop into a male. However, there is a gene on the X chromosome that is called the androgen receptor gene, the AR gene, and its job is to make receptors for testosterone. If that gene is mutated, then even though the individual is making testosterone, there's no receptors to receive that testosterone, and therefore the embryo develops as a female. So now we have an individual whose genotype is XY, but whose phenotype is female. So how do we take these X chromosomes and condense them down so that they become bar bodies? There is an area of the chromosome called the XIC, stands for the X inactivation center, and this is where inactivation begins, or condensation begins. And this whole area, this whole diagrammatic area here is the XIC. You can see that there. And there are a couple of important areas. This gene here is called XIST, and if you look on this one, this is TSIX, or if you look at it that way, it's also XIST but backwards. And that's because this is a reverse gene to there. So that gene is going from right to left, this gene is going left to right. And there are some non-coding reverse RNAs involved that control the expression of these genes. So if you have a female that has 2 X chromosomes, and you can randomly inactivate one of those X chromosomes, then it's possible that identical twins could actually be non-identical. So for instance, here are 2 identical female twins. They both have X chromosomes, and in one twin, let's say this chromosome on the right was inactivated, they will express whatever genes there are on this chromosome on the left. On the other twin, let's say that the other chromosome was inactivated, so they will express what proteins are made from the other X chromosome. Well, if this one is, say, big A, makes big A protein, and this is the recessive little a, makes little a protein, then of course you'll have different expression patterns depending on what genes you're looking at. We can see this in an individual by looking at two cases here. This is called a calico cat. You may have heard of these. These are calico cats. They're always female because they have 2 X chromosomes. And somewhere over their body, sometimes this left-hand X chromosome is inactivated, and sometimes the other X chromosome is inactivated. And depending on which one is inactivated in the fur, in the skin cells producing the fur, gives you this mottling of color. So we can have this black color here produced by one X chromosome, and the brown could be produced by another X chromosome. So you have this calico looking cat. In humans, there are some sweat glands that are produced on the X chromosome, and if you happen to be heterozygous, so you have big H, little h for these sweat glands, and if the big H chromosome is shut down, then you have the recessive phenotype where you do not have the sweat glands, and that's this yellow area here. On the area where you do have sweat glands, you would have the dominant H on the other X chromosome being expressed. And so human females, can also be a mosaic of gene expression due to this inactivation of random X chromosomes, these bar bodies. So traits can also be influenced by one's gender, and these not necessarily influenced by in the same way. For example, sex-influenced traits they are autosomal genes, but they can be dominant in one sex but recessive in another sex. So even though a male and a female may have the same genotype as far as this particular gene is concerned, they may express it differently. Pattern baldness is an example of a sex-influenced trait. It acts as an autosomal dominant trait in males and an autosomal recessive trait in females. And in this case, this male has the dominant trait and he is bald, as is his siblings. There is another interesting way of controlling gene expression, and that is called imprinting. Imprinting happens if you have one chromosome from your father that is methylated in one fashion, and your same chromosome from your mother is methylated in a different fashion. What methylation does is it changes gene expression. So let's have a quick look at this. If we have two chromosomes, this particular one happens to be chromosome 15. There's a gene on chromosome 15 called the SNRP-N gene. This chromosome on the left, shall we say, came from your father, and this chromosome on the right came from your mother. The methylation patterns, methylation being CH3, the methylation patterns on these two different chromosomes are different because they came from two separate people with different genders. If you have a mutation in this SNRP-N gene in the father's chromosome, it will produce a different phenotype than if you had exactly the same mutation in the chromosome from your mother. And this is what it manifests itself as. If you have a deletion, this mutation, in the father's chromosome, in the paternal copy, you have a disease called Prader-Willi syndrome. If, however, this deletion occurs in the maternal chromosome, this one over here on the right, then it will give rise to a different disease called Angelman syndrome. These are pictures of individuals with Angelman syndrome and Prader-Willi syndrome. Angelman is known as the happy puppet because children always seem to be smiling and seem to be having a good time with Angelman syndrome. Prader-Willi syndrome, on the other hand, is characterized by overeating and consequent obesity, amongst other things. This diagram is a karyotype, so here are all the different chromosomes, and they've been stained for particular genes. This is chromosome 15 here, and here's another chromosome 15, so one's the mother, one's the father one. And they've been stained for a couple of genes. In this case, one gene is PML, just to show you where it is, and here's the PML gene on this chromosome 15. And over here, here's the chromosome 15 PML on this one. But if you look at the other piece of yellow, this is the SNRP-N gene on this chromosome, so that's there, it's working just fine. But the SNRP-N gene on this other chromosome is missing. There's been a deletion on chromosome 15. And so depending on where it came from, and in this case it comes from the father's chromosome, so this chromosome came from the male, this will give you, in this case, Prader-Willi syndrome. If this chromosome was okay, and this SNRP-N was missing, then you'd have Angelman syndrome. Now this imprinting is not permanent. In each generation, the imprinting is erased and the whole gene is re-imprinted with your mechanisms. And this happens during gamete formation, in other words, during meiosis, or very early in development of the embryo. And so imprinting is an epigenetic change because you're messing with methyl groups. These methyl groups will change the expression of genes depending on where they are added. And this is therefore not related to your genetics, it is related to your epigenetics, something beyond the genome.